objectives for this particular one are to set up a serial dilution and describe why they are used, to calculate CFU per milliliter from colonies on an agar plate, given a dilution factor and a volume correction factor, and to experiment and interpret your results, and to provide examples of why bacterial growth rates have to be measurable and characterized. So we're going to talk about these concepts. We're going to talk about serial dilution, calculating CFU per milliliter with dilution factors and volume corrections, and that's going to be most of what we continue to talk about here. So there's some theory down here. I don't really want to go into it too much as it's kind of confusing, but we'll hit on a few things. So bacterial growth, bacteria, as you know, goes through binary fission to create two identical cells when it undergoes generation. So two cells, or one cell, will become two cells will become four cells will become 8 cells, and essentially every generation you will double the amount of bacteria that you have. And that's represented as a number like this. So 2 to the 0 would be your initial bacteria, and after 3 generations you'd have 2 to the 3, or 2 times 2 times 2, so 8 cells total. After 4 generations, 2 to the 4, you'd have 16, 2 to the 5, you'd have 32, so on and so forth. So that's just something to remember about how bacteria grow. And then they talk about colony forming units. So colony forming units, thinking back to how you guys plate bacteria and read bacterial plates. You guys did some work with Salmonella and E. coli and some of your other simulations. So each of those little colonies that form on your plates are considered colony forming units. And they're an estimate of the number of viable bacteria or fungal cells because they're actively growing. If you measure the concentration of bacteria in a tube just with a spectrophotometer, which I believe we talked about in the last lab, it will measure both the living and the dead cells because you can't separate the two of them when they're mixed in solution. So the colony forming unit on a plate will only measure the living bacteria in a sample. And that's kind of an important distinction when doing microbiology experiments. So for their example here for calculating colony forming units, and we'll look at this on the Jamboard here in a second, but they suppose that you had 25 colonies grow on a plate, and you plated, the original amount that you plated was 0.1 milliliters. This is kind of the essential equation to know. It would be CFU per milliliters is colonies over the volume spread. So it would be 25 colonies over 0.1 milliliters. And that would equal 250 CFU per milliliter. The next thing that they're going to talk about here is one more extension of this equation. So this is the basic equation to understand. The next extension of that equation is using what happens when there is, I think what it said up here, a volume correction factor. And you get this correction factor when you do a serial dilution. But let's look at colony forming units first on the Jamboard. So this is what it's actually going to look like in Labster, the difference between the two. So let's see. So on the left here is what we just talked about with that equation, which is your number of colonies, which in Labster you're going to be adjusting up and down, and the volume used for spreading. So when you're literally plating the bacteria, you need to know how much you're adding to that plate and spreading around, and that's going to be this value. So when there's a volume or a dilution factor or a volume correcting factor, a dilution factor is an extension of this equation where you have your number of colonies divided by your volume used for spreading, and then you would multiply by a dilution factor. And that's what we're going to talk about second. But first, let's do an example with your first one here. So say, for example, I'm going to use this. So say, for example, this is a tube full of E. coli that you've grown. All these happy little E. coli, you've grown it for 24 hours. So it's a cloudy solution, and you want to know how much of that bacteria is alive on your plate. So essentially you would take a pipette and you would pipe it out of here onto your plate, some amount. It's usually about 0.1 milliliters. It's usually a fairly small amount. And then you would spread this on your plate with your glass spreader after you sterilized it. You would incubate this. So this took 24 hours to make. Then you would plate it, and then you would incubate for, I don't know, whatever you're growing has different times that you would need to incubate it, but maybe say incubate for 24 hours at 37 degrees C. So there's always a time and a temperature, and you would get spots forming on your plate. So if we counted these spots, I believe there's 8 of them. So this would be 8. So I put our little equation down here. So it's 8 colonies, or CFU. It would be 0.1 milliliters, because that's what we used to plate it. So 8 divided by 0.1 would equal 80 CFU per milliliter. And that is how you would set up that equation. So going back to this original one, what do we do in case of serial dilutions? So what I set up here is to explain a little bit about serial dilutions for you guys. Maybe. So imagine we had that same bacterial colony, 24 hours, E. coli, lots and lots of cells in here. And if we plated that directly onto this plate, we would probably, instead of 8 colonies, we would probably end up with like more than 1 million colonies. Some ridiculously large number that we wouldn't actually be able to count on this plate. Because it is so concentrated and there's so many bacteria, after they double and double and double and double, they're growing exponentially. So you're going to have a lot of bacteria. And the goal of a serial dilution, so let's write that up here. So the goal of a serial dilution is to reduce the number of bacteria on the plate to a countable level, so that you can estimate the original amount in the stock solution. Whoa, I can't spell. So if our stock solution, so let's call this our stock solution here. What we're going to do is we're going to set up a series of tubes, and it's usually going to be quite a few, but for simplicity's sake, I'm going to do 3 here. 3 tubes. I I don't like that tube, but he can stay over there. And each of these tubes is going to have 9 milliliters of just, let's call it H2O, but it's probably going to be some sort of a broth. And what we're going to do is we're going to pipette. Let's change colors here. We're going to pipette like we did on the last slide, 0.1 milliliters, or no, not 0.1. Sorry. We're going to pipette 1 milliliter of the stock solution into our first tube. And what this is doing, if you have 1 milliliter of stock and 9 milliliters of diluent, this is basically a 1 to 10 dilution. So 9 parts water, 1 part bacteria. And so your amount of bacteria in this tube is going to be a factor of 0.1 or 10x less than the bacteria in your stock solution. You would then vortex the sample to really mix it well, and then you would do the exact same step. You would transfer 1 milliliter of bacteria from this tube. So this tube now has, let's draw that a little bit better. So this tube now has 10 milliliters in it, and you would transfer over to here. And now this tube now has 10 milliliters in it. And it's also a 1 to 10 of this tube. So now your number, your dilution in your second tube would be 0.01, because your decimal point is moving to the left each time by 10, because this is a 1 to 10 dilution. And this would be 100 times less bacteria in this tube than your original tube, or 10 times less than your second tube. And the very exact same thing would happen. Green. Sorry for those people who are colorblind. We would add 1 milliliter into our final tube from our third tube. This would be now at 1000x. Just add another 0 there, or 0.001 times less concentrated than our stock solution. Or it would be a dilution factor of 1,000 that we'd have to multiply by to get the actual number of original bacteria. So if we didn't want to plate our stock solution because we automatically knew that it was going to be too much bacteria, what we would do next is, after we set up these 3 tubes of bacteria, we know their dilution factors, which are these numbers. We would plate from tube 1 that number that we used on the last slide, the 0.1 milliliters, onto this plate and spread it around. I should have used red on that one because the red's going onto this plate. The blue is going onto this plate. So we would plate 0.1 milliliter of the blue tube onto this plate, and green would go onto this plate. So from our tube, we would add 0.1 milliliters onto this plate. So each of these is 10 times less concentrated than the previous one. This one's 10 to the minus 1, 2, 3. This one's 10 to the minus 2. This is 10 to the minus 1. So what would we expect to see on these plates after they incubated? I didn't actually prepare this slide, but let's make a new slide. So what would we actually expect to see if we grew these for, I don't know, 24 hours. On our first plate, if on our first plate, this is completely hypothetical, if on our first plate we had 300 bacterial colonies, what would we expect to see on our second plate? We would expect that there's 10 times less bacteria in our second tube than in our 10 to the minus 1 tube. Or that's 1. This would be 10 to the minus 2. And so we would actually expect, if there was 300 on this one, we would only expect there to be 30 colonies or CFUs, colony forming units on this plate. And again, if we went to here, we would only expect that there was 3. This raises an interesting question, because what happens if we actually had a 4th tube and diluted one more time? We would actually expect that there would be less than 0 colonies or CFUs if we did a 10 to the minus 4 in this case. So this plate would actually be kind of useless, because there wouldn't be any bacteria growing on it, because it would be so diluted that we wouldn't be able to see anything growing. So the optimal range for colony growth is going to be between 30 and 300 for the best range to count in. So if you're asked to pick the best plate to count, you want a plate that is hopefully within this range to be most statistically correct. So let's say that we went with this plate with our 30 colonies, because it's easier to count than 300 colonies. So if we had 30 colonies at 10 to the minus 2 here, and we know that we plated 0.1 milliliters onto that plate. Let's go back to our original here. So we had, if we had 30 here, we know that we plated 0.1. And this number over here would be 10 to the 2, because this was our 10 to the 2 dilution. Again, this was our blue plate, our second plate. It's not moving. Our 10 to the minus 2 plate had 30 colonies on it. So this would be 10 to the 2 as our dilution factor. So our equation would be, and you guys can do this on a calculator, it would be 30 divided by 0.1 times 10 to the 2, which would be 1 with 2 zeros, which would equal, let me break out my phone calculator, because I do not guarantee that I could do it math in my head right now, times 100 is so instead of a plate with 30,000 bacteria growing on it, which is what we might have gotten if we did our stock solution, our plate has 30 colonies on it in a countable range, and we can calculate that this was the original concentration of our stock solution. And that is what you would need to set up this equation if you were asked a question about calculating with a dilution factor versus just calculating with your volume and your number of colonies. So for this one over here, it would be basically assuming that we used our stock solution. There would be no dilution factor, and it would just be 50 over 0.1 equals whatever that is, like 5,000. 500 CFU per milliliter. And you always want to make sure that you're using units. Otherwise, the numbers don't mean anything. So, so long as you have units there, people can understand what you're talking about. If you just throw out 500, it could be 500 jelly beans in lemonade or something like that. It could be something completely different than what you're talking about. So make sure that you're using your units when you're talking about these things. Great. So that's if you go back to those documents, they talk about serial dilutions in a kind of a strange way. It's worth reading through and worth understanding, but this is really what you need to understand to do these calculations with bacterial plates. So if, for example, you were given a question that gave you the number of colonies on a plate, the dilution factor, and the amount plated, can you actually set up that equation? So what we were just talking about, you would have your number of colonies, so you would have 25 divided by your amount plated, 0.1, and you would multiply that by your dilution factor. 10 to the 3 would be 1 with 3 zeros, so 1,000. And that would be from your plate that was 10 to the minus 3. And that would equal, I think I already did the equals on this one, 0.1 times 1,000 equals 250 1,000 CFU per milliliter. So that is what that equals. So just know if you are given these numbers, know how to set up this equation, and potentially if you had a calculator, you could do the actual math. Great. And then the other thing I wanted to talk about, the documents and the simulations will go into your bacterial growth phases. So I did want to touch on the 4 bacterial growth phases that you'll see when bacteria are growing. I believe this is the second simulation that you'll be doing. So there's 4 phases. There's phase look something like this. Then you have the exponential phase, which will look something like this. Then you have a stationary phase where the bacteria, so the bacteria have to get used to the nutrients they're in. Once they're used to the nutrients and the location they're growing, they're going to start doing that binary division, and they're going to start growing exponentially. Once they start running out of nutrients in their broth, they're going to go into a stationary phase. And then finally, if they use up all their nutrients or get old and die, you will see a decline at some point. So these are the 4 phases, 1, 2, 3, and 4. And they will look like this variously. You can have extended leg phases. You could have your exponential phase closer over here. You could have an extended stationary phase. But those would be the 4 phases to know of your bacterial growth. And just know that they're getting used to their environment. They're growing happily. They're running out of space. They're running out of material. They're producing waste in their environment. Something is slowing them down. And finally, they are dying.